All right, hello. Welcome to this tech talk today on pharmaceutical spray drying. I wanted to start off by understanding why we would consider this. The main reason is that 90% of the compounds coming through development now are insoluble. I've heard 70%, I've heard 80%, but in reality it's roughly 90%. The nice thing about this though is it's dictated by the development classification system, and in order to qualify as one of those 90% of insoluble compounds, your drug needs to be insoluble across the whole aqueous pH range from 1 to 9 at the highest dose. So for example, if you have a 200 milligram dose and you're using 500 mils as your volume, you need to be soluble at 400 micrograms per mil across pH 1 to 9. Most compounds aren't, and so they qualify as a DCS class 2 compound. But in reality, they sometimes behave a little bit better than that in vivo, so you're always trying to figure out the easiest way to deliver the drug. In some cases though, you really need an enabling technology to move that forward. So for compounds that fall within the DCS class 2B category, which means they're going to be solubility limited, spray-dried dispersions will give you a very good advantage over other types of delivery systems. This is the question you cannot fail. Somebody needs to answer this one. Which PK profile do you want? The blue one I heard whispered. I think it was not with a lot of confidence, but in reality he's right. For difficult to solubilize compounds, that's a great PK profile. That's what you're trying to shoot for with the enabling technology. Spray drying has been along for a long time. It's been used in the food industry much longer than it has in the pharmaceutical industry. So powdered milk, other types of commercial products like coffee are spray dried, and they've been spray dried that way for a long time. So it's nothing to be afraid of. The one concern with it though is it does generate a very high energy dosage form, and so you have to be able to stabilize that dosage form and understand how you're formulating it to move it forward into the development path. The other nice thing that it does is it creates a supersaturated media that allows for increased absorption in the gut and in the small intestine. The final dosage form that you present will be a tablet or a capsule, so it's also very nice that you have a solid dosage form you can take forward. It's a nice option in combination with lipid delivery systems, depending on which one works better for your BCS2B compound. You take one or another forward. In its simplest form, spray drying is simply taking a crystalline drug, which to a medicinal chemist, that is beautiful. It's a very nice crystalline material, probably highly pure, but to a formulator, it looks like something that's probably insoluble, that's going to be difficult to work with. And in this case, this one has a high melting point, somewhere in the 200 degrees range or so. And so it just doesn't have good aqueous solubility. So if you can dissolve that compound with a solvent that has a low boiling point, such as acetone pictured here, and combine it with a polymer, you've created a solution in an organic system that then, with some processing, will actually result in a nice powder on the back end. And this is where it gets interesting. The processing is pretty cool. So you're doing a spray drying work, you're pushing it through a cyclone with a lot of airflow. It's creating a high energy system, a high energy dosage form, and as Dorothy might have said in The Wizard of Oz, you're going to get a magical outcome. What you're hoping for is a nice mixture of the drug and the polymer intermixed together in a very homogeneous way, and so that there's not any recrystallization happening. The resulting powder comes out as a relatively low bulk density material, and I'll explain a little bit more how you process that later. This is spray drying in its simplest form. This is a model of a very small scale spray dryer, a Buki B290. You have your feed solution, which is the solution we just made it for, which was drug, polymer, and a solvent. It's going through a nozzle that is atomizing the drug into the solution into very small droplets, co-current with warm air, and that air is drying those particles within a second of time. And so kinetically, you're not allowing the drug to recrystallize before it forms a amorphous dispersion. The temperatures for the inlet on a spray dryer are normally around 90 degrees C, depending on the solvents you're working with. And down here at the outlet, at the bottom of the cyclone, you're going to get a shriveled particle that has a density of about 0.2 if you're doing it properly for a solid oil dosage form. And that material is going to be relatively dry. It'll have some residual solvents with it. The outlet temperatures down at the bottom down there are around 50 degrees, sometimes a little lower, 45 to 50 degrees C. So you do need to be concerned about a little bit of the thermal stability of your compound. That's really not that elevated temperature. This is an image of the Buki B290 small scale spray dryer. This is where you would start development work. You would eventually move it to larger scale spray dryers like mobile miners or PSD1s or PSD2s to do large scale clinical and commercial productions. The key to developing a dispersion is generating a supersaturated media. And so you see some dissolution curves here on the right. The x-axis there is the time in minutes, and the y-axis is the concentration in micrograms per mil of the drug in a biorelevant media. So the way we do this study is that we start with 30 minutes in an acidic media, so 0.1 normal HCl, and then at that point we quench it with FASIV solution. So you're now at a more neutral pH with surfactants involved, and you're measuring what's happening to the drug over time. And in the beginning here, you went from a crystalline drug that had a solubility of 10 micrograms per mil or so to something that was an order of magnitude of 3 mg per mil. It clearly is not stable at that concentration, And so you're hoping in vivo what's happening is you're getting stabilization with the salts in the gut, or you're getting flux through the lipid membrane and you're getting absorption, and so it doesn't get a chance to recrystallize out. In development, we've got a lot of polymers listed here. HPMCAS, PBPVA, PVP, methacrylate. These are common polymers used in spray drying. You'd want to screen most of them, and you can do some modeling up front to determine which ones might work best. When you get these sort of profiles in vitro, you would want to sit down and decide which ones you want to take in vivo for an assessment. And looking at these 5 curves we have here, I would recommend taking 2 in. One would be the PVP solution, the dispersion, which gives you the highest Cmax. And the other one would be the HPMCAS dispersion, which gives you the longest sustained solubility down here. Those 2 in vivo, you're not quite sure how they're going to work, so I think you want to put them both in the animal model that you're choosing and take that forward and see what you get from a PK profile perspective. As I said earlier, the material that comes out of a spray dryer, it's beautiful in the sense that it's going to give you better solubility, but from a downstream processing step, it's a little bit bulky, it's a little, the flow material isn't very well, so you need to have roller compaction typically used to process that material into something that can then be put into a capsule or a tablet. Not all roller compactors are created equal. The roller compactor we prefer to use is the Gerteis roller compactor. It allows for feed of very poor flowing materials and getting very consistent ribbons and granules. And so in this process, the material is charged to the top in the hopper. Right by step 1 there, it's horizontally fed through with an auger into the area where it's going to be fed into the nip region of the rolls, and then you're going to generate a roller compacted ribbon there. In the intragranular portion of the ribbon, you're going to have a filler and probably a disintegrant, maybe a lubricant, along with your dispersion material. And then that material is going to be milled all in the same device, and it's going to come out as granules that will then be processed with extragranular material and compressed into a tablet or filled into a capsule. So at the end of the day, the purpose of doing a dispersion is to see if you're going to have an advantage in vivo over other delivery technologies, other formulations. Something we do here at Catalent is an Optiform solution suite where we look at agnostic approach at different types of formulations. Spray drying is just one of them. And so you would take that data in vivo and you would generate these types of PK profiles. And what we like to do is sit down with the client after that data is generated and talk about what is their ultimate goal for this program. Some clients are in early stage, they're maybe in phase 1. They don't want to put a lot of extra effort into the formulation, so they'll look at this data and they'll see the dispersion looks great, but they may be able to get away with something that's a micronized material with a surfactant for their purposes in phase 1 and phase 2. In other situations, that's not adequate bioavailability, and so we would take the dispersion forward. But you want to be able to be flexible with how you deliver that next solution, which is really the phase 1 or 2 dosage form that you're developing for your client at this stage. Thank you very much. Appreciate your time.